Yes, this is Leif Busch from Permaculture Farmers. I'm going to tell about the establishment of regenerative farming or regenerative agriculture and permaculture design. And this is sort of the whole picture. We move on. So the first thing is what I call the agriculture biological cycle. It is actually plants, animals, and soil, and this exists in most farms where at least they have animals. Of course, it doesn't exist if they're using artificial fertilizer. So the one we are working on developing here is regenerative agriculture biological cycle. And basically, it is the same. What the point is put on having this extra. We have photosynthesis, and then we have this extra output in here, and the photosynthesis is going directly into the soil. And the idea is to develop carbon, just much biology in the soil as it is possible. I think this cycle is the basic of farming, and I don't really believe in any kind of farming where we don't try to build the complete biological cycle or nature's biological cycle. Okay, the situation on almost all Danish agriculture, and this is just because I'm in Denmark, I say Danish, and then because I know the Danish farm and soil quite well. Very low content of humus and organic matter. Heavy mechanical soil treatment, often every year. Reduced root zone, traffic zone, plow zone, often anaerobic condition below plowing depth. The soil contains disease-causing bacteria and fungus. There is often no biological cycle in the soil. It means that cycle we see before, regenerative cycle we see before, it doesn't really exist. Spread use of slurry, fertilizers, and chemicals. Slurry is of course manure, but it's mixed in with the urine, and it's stored in often 3 or 4 meter deep tanks that are anaerobic, and it can lay there here in Denmark for well up to maybe 9 months, something like that. Contamination with drug residues on the field. Widespread need for irrigation on sandy soils. Sometimes, some years when we don't have too much rain in the spring, crops are just coming out of the ground, and then we have to start irrigating. Of course, it is because of this problem here with very little soil growing. And this is one of the things we have to solve. Monoculture year after year. Okay, so what is it that we can do with this thing? How are the problems solved? And we solve the problem by using the soil food web method, as it is trained on soil food web school by Dr. Elaine. And this is a method where we look at the soil with a microscope. So we look at all the soil, we look at we're making compost, we look at it and so on. So we follow the whole development of the soil simply by looking at a living soil, or at least making it living. So this is how it looks when we look at a typical conventional farming soil. This is just bacteria and nothing else. And normally it is a disease bacteria because the only thing that can actually grow in this soil, the only thing that can grow in this soil is the bacteria that are living on our crops. So we are sort of promoting bacteria we don't really want. Next thing is fungus, and these are omocytes, fungus diseases, and those look like this in the soil. This can be early blight, and it can be mildew or late blight in potatoes. So these are the diseases, and again, we are promoting them by having monoculture crop and no biological life and biological cycle in the soil. The earth does not lack nutrient, it lacks life. And that life should be fungi, bacteria, and protozoa, all useful, not all these disease ones. This picture here is put in because we have these circles around here, or spots, which is very green. And some scientists were working on this thing, wondering why are they there? And since it's very, very different from the surrounding landscape. So they find out of that 2,000 or 3,000 years ago, maybe the farmers were having cattle, the animals here in big crawls, and maybe they get up to 1 meter, half a meter or 1 meter of manure laying all over this place here, which are then being composted here. They leave the place and just move to the next place. And now, since then, animals come in every year. The game come in every year to graze in these spots. The game know, of course, this has a very good grass, so they come in to eat this thing. But this is sort of to show that we can restore, or store the introduced bacteria, fungus, and protozoa to the soil, and then have this thing running year after year, if we keep this biological cycle running. Some of these spots here are found to be over 3,000 years old. I put a link down here just for people to go in and check it themselves, because they might think this sounds a bit unbelievable. But this is how nature is. If you give it a chance, it will go for it. Now, what can we do about it? I mean, what can we do to actually sort of get this situation here in our farms? A very productive biological cycle is running here. So this is the soil food web, and this is the system that are sort of found out by Dr. Elaine, and it is very, very important for sort of all farmers. And I think it is very important for the future of farming. What microorganisms must be present in the soil? The whole microbiological food network, the soil food web, we have it here. I don't want to get into details here, but this thing starts with sun, and then the fungus and the bacteria especially, they live on or from, exudates from the roots and live on organic matter. And there's some nematodes also here, root feeders, which we know very well from diseases here in Denmark. Potatoes especially, we have a lot of problems with it. The next link is that this level here eats this level there. Actually, it is so that I very much see bacteria. If you compare it to something we can see on the surface, then I'll compare it to grass up on the surface, and maybe the fungus to dicotyledons, all the other ones. So these are really the two, especially grass, which are growing, or bacteria which are growing the lot of the nutrition, picking up a lot of the nutrition we have in the soil. So those ones here are eating them, and the waste from those ones are then going to the roots of the plants. Waste of those ones here are inorganic fertilizers, because they eat sometimes somewhere between 10 and maybe even up to 60,000 bacteria in a day. So every time they just eat it to get enough for themselves. A lot of waste are going out as inorganic fertilizers. The next one, to keep this ones here in balance, we have the next one, which is then of course eating this one. Now we are up on the surface of the fields, and out here sits of course Mr. and Mrs. Denmark with some vegetables, some pork and beers and whatever we are growing here in Denmark. So this is the whole food web we grow, and this what is needed to actually make a farming regenerative. This is what is the minimum. So they look a little bit like this, not much talk about this. Beneficial fungus typically look like this one here. They have a dark color and some septums across. We have a lot of testate amoebas which are eating the bacteria. We have amoeba, chaos amoebas, which are also eating lots of bacteria. This one here is about to split into 2, even 3, I think, a little bit later here. Then we have flagellates, many different kinds of flagellates. We have ciliates, and ciliates has this interesting angle on it that if you see a lot of it in the soil, you will also know that this is because the soil is anaerobic. It has too little oxygen. And so this is sort of a blessing in disguise, because you can look at this thing and then you can see, I have a problem. But as few as possible of these ones, but they're also eating. They're at the same time they're beneficial. They eat, again, maybe 20, 30,000 bacteria in a day, and waste will be available as inorganic fertilizer to the plant. This is a bacteria eating nematode. We have plenty of different kinds of nematodes, actually root feeders, which are not the ones we really want. And then we have fungal feeders, bacteria feeders, and predators, which are primarily eating the root feeders. A little example of how it looks in the soil as compared to this one here, these two. Then the soil, when it's alive, it looks like this. It's here we see a secosaur, testate amoeba, something that might look a little bit like a fungus, or maybe it's just a piece of, or it actually have a little bit of septum. So we say it's fungus. Otherwise, it will just be a piece of plant material. Some fungus here, and over here we have this is a piece of antenna from an insect. Definitely some fungus here. We have something here. I don't really know what it is, as it's sometimes difficult to see in the picture, but it looks a little bit like fungus, even though I think it's a plant material. And again, some fungus, maybe an amoeba cyst here, definitely some amoeba cyst in this one here, and yeah. Here over here in this one, I don't know even why I have it here, but definitely it has some humic acids, and this might be fulvic acids, and this is what we want to build up in the soil. So some of the insects here, microarthropods, which are also doing a lot of good. So all of this stuff will start developing in the rhizosphere of your plants when you add what we call biocomplete compost. We will get to how we make it and all this, but this is what it's all about. And this can be added in the form of actual compost, and maybe as an extract, and even as a compost tea. So the microorganisms will develop in the rhizosphere of the plant root, and simply the plants are promoting the microorganisms they want to grow inside the rhizosphere, and they do that by giving out exudates, which are sugars and whatever the microorganisms might need. So they develop this thing for their own benefit. It is exactly like we do farming, really. The plants are actually doing some sort of microorganism farming inside this rhizosphere here. And so it's when we are growing crops and pigs and chickens and cattle or beef, the same way the plants are growing, especially fungus and bacteria in here, and that is promoting the growth of the microorganisms that are eating the fungal and bacteria and so on. So they get the whole biological cycle. The whole of this biological cycle delivers nutrition directly from mineralizing the sand, silt, and clay. And this is the very, very important thing to understand here, and the very big difference from conventional farming, where we are adding all the nutrition in the form of either slurry or inorganic fertilizers. Mycorrhiza can be formed here in the rhizosphere and extend the roots out. And we'll get back to this thing, but this is extended like like 800%. A little bit about what's in the soil. This mineral is in the soil, sparked 2003. And the headline here is, we have all the nutrition we need in the soil. So if we can get, and by soil here, I'm talking about sand, silt, and clay, as the actual texture, as we call it in the soil. Sand, silt, and clay contain all of these nutrients, and of course many more. This is just the ones that have been tested here. All nutrients that a plant needs is inside the soil. That's the takeaway on this one here, and being made available by this bacteria and fungus delivering to the plants. So how does it look here? The nutrition pool. So this is sort of a picture of the figure we see before. This is the whole thing. The blue box can say, you can compare this thing if you have a handful of soil, then you have a lot of nutrition there. And it's not a dead soil. So the big blue box is the whole total everything nutrition here. And then the bacteria, fungus, and protozoans and microbes, they work on it, and you get into this smaller box here. I mean, they are pulling it out of the surface, and then the small one, and this is the sort of dissolved in soil solution potentially available to plants. So the plants are only getting the nutrition from a soil solution or a water solution inside the soil. And this is a very, very important thing when we look again here. All of this is the blue box. And then come the bacteria and make it bacteria and fungus and protozoa, the whole biological network. Web, soil food web is coming to make it available to the plants. This little pull down here, soil solution, this is the only thing we measure on in conventional farming. And this is what makes it a bit confusing to compare. So we measure on this fraction of the available solution, the available fertilizers in the soil. Okay, so the way we look at it, we look at it like this: soil, plants, animals. This is the conventional. We start with the lands, and this is right. We do start with the lands. It's a little bit like the circle I started with, which is saying soil, plants, animals. It is not wrong, but the point is we have to look at the soil as something that is alive and not just a dead and constant material we can use for whatever we want, and it will still remain the same. It's not the case. We are spoiling it. That's the main issue right now. Photosynthesis, green plant light, CO2, water. The soil, the look at soil should really start with photosynthesis. And then we move on with all the plants growing, the rhizosphere, whatever we talk about, roots and associated microscope, the soil food web coming in here. And then comes soil, and this is biologically active soil. So it's a whole different situation, a living soil, not a dead one. We should not forget one thing, that the soil is actually the stomach for the plants. You can compare the soil to our stomach. The soil makes the nutrition available for the plant roots, and the soil biology is the same as what we might have in our own stomach. Often, actually, the same kind of microorganisms are in our stomach. Okay, what does it take to reorganize agriculture into regenerative agriculture? Well, then the answer is here. The soil biology, soil food web must be regenerated so that the soil contains the entire biological network, which through plants and photosynthesis is the basis of all life in the soil. Wow, it's almost like listening to the Bible. So there's 3 steps here to do that. Biocomplete compost must be grown. Don't forget I say grown, must be grown. Okay? We grow it like we grow crops, pigs, and cattle. That's how we grow this thing. And it is difficult to learn on. It depends on experience. You must have this thing in your hands to learn how to do it. So compost is not just biocomplete. It's not a fancy word for compost. Most compost is actually not good. I have been looking at lots of compost around in Denmark, wherever I come across it. And I've never found anything that is actually biocomplete. I find something that are very much filled up with this humus seeds we talked about before, and something called klimavenlige bacteria, which are chains of bacteria, and probably a lot of other bacteria also that are not beneficial at all. So biocomplete compost must be grown. Biocomplete extract must be prepared. And that's because the biocomplete compost, you actually prepare extract like you make coffee. So or tea. So you take out a portion and then you are washing out all the microorganisms inside the biocomplete compost. You wash it out and then you will have a biocomplete extract. And you have to put in the amount of compost depending on how much bacteria and fungus and other microorganisms are inside. Okay, next one, biocomplete compost tea must be grown. Again, we take here something we have to grow, and it's very difficult to do this thing right. I've been struggling a lot with it. Biocomplete extract must be prepared, and that means we have the extract, which is now ready. This is a liquid form, and we then want to grow a tea, which means we put it inside some machinery we will get to, and then we keep this thing running, and we give it actually some fodder. We give these microorganisms some fodder, and we give them fodder according to what kind of microorganisms we want to grow in this compost tea. Okay, so this is the 3 steps. We start with step 1. Biocomplete compost must be grown. So here we have a stack put up in some layers. It's not very important. We see this thing all the time on YouTube that is put up in some layers, but it's not very important to put it up in some layers. What is important is that we have the right amount of different ingredients inside. So this thing here is more for YouTube than it's for anything else. I think the best thing is to mix it up in one big pile and then put it all in that mixture. Biocomplete compost is grown by mixing fresh, green, nitrogen-containing and dead woody plant materials according to a seasonally adapted recipe. Okay, that means the temperature goes a little bit up and down here in Denmark, and it's the same thing in other places. So we would change how much nitrogen rich material is actually inside. At the same time, if you grow it in the winter here, you have a lot of problems in getting fresh green materials. Actually, you will not get it. So there's different ways of doing this thing. So this seasonally adapted recipe is very important. You might have success with growing after a certain recipe in the summer, and the same recipe will fail completely in the winter. But the basic thing here is that we will have to look into and make sure that experience only will learn people to adapt to this recipe. Diversity is important, as much different plant materials as possible. Biocomplete compost can be spread on a bare field and harrowed down. And it can also, some people would also spread it on a bare field. In the case of a planted field, it is spread over the plants, the crops, on a cloudy and rainy day. So you can spread it out. It is fine, very good if it's raining. You can imagine this thing being washed down into the the roots of the plant. Yeah, a little bit more about this thing. The quality is paramount. So we really need to have the right numbers of microorganisms inside here. We will get to the microorganisms a little bit further down. Here I can take it immediately. The bacteria have to be 135 microgram per gram. The fungal 135 microgram per gram. FB ratio, this is fungal bacteria. It has to be equal or greater than 0.3 to 1. And yeah, very, very important. We have the producers, 10,000 per gram of compost. These are those many different ones, different amoebas, flagellates, ciliates, or yeah, different kind of things running around. Beneficial nematodes, 100 per gram of compost. So if you count on your microscope, you see, you have to learn to know when you count the nematodes, you can actually finish your sample. If you don't see any, the compost is not going to be okay. It's very important. Ciliate less than, and this is because if you grow compost and make compost or compost liquids, you are not going to make this thing with a lot of ciliate inside, because then it's anaerobic. And at the same time, you will be growing especially actinobacteria inside. So it has been too hot. You have to try to restore. Now back to this one here. When growing a biocomplete compost, it is important to control temperature and external influences. So wind, frost, rain, everything, we try to sort of regulate. It's not that the compost cannot handle the weather conditions. It can. It will adapt to it, but it will adapt like the rest of nature with the way that this thing is working. The stack should be turned at least 2 times at 65 to 70 degrees Celsius. So all material has been in the center of the stack. So it goes like this. We take off the top. They say this is the first time it's stacked up, and here it is laying, and now it is 70 or 65 or 70 degrees inside. We take the top off and put to the side, and then we start putting the net on the other side, and we start sharpening it over. And when you get sort of up to about a little bit 1/3 full, you take the top stack and the top part and put inside into the middle, and then you continue up to the top with the ones that was in the bottom. So this is the first time you start it, and you do that 1 more time. So this is 1, 2, 3 times, so you get all in the middle. Temperature must not exceed 70 degrees Celsius, and the stack must always be aerobic, must always have air. So if we get up high in temperature, the problem is that the bacteria will use an awful lot of oxygen, and you can actually make it anaerobic in a very, very short time. And then how long time does it take? It takes between 3 months and 14 months to make biocomplete compost, depending on the condition and the quality desired. And this is, I can make it on 3 months when it has to be biocomplete. I can even make it sometimes on 4 weeks, but it's really down on the limit. And then it's only in the summer, really late summer, when you have humid, little bit cooler weather and really good conditions for growing fungus. 3 months is what I prefer to do, and 14 months we'll get to why I think it takes that long time really, and this is what I prefer to use to make compost in over 1 year. Yeah, and then biocomplete extract must be prepared. It is now we have the biocomplete compost, as we talked about, and I use 5 to 10 kilo biocomplete compost in a 1,000-liter tank, and we blow air inside for 30 to 60 minutes. Actually, we just blow air inside until this net, 400 micrometer net here, is empty for small things. Only what's left in it is now the big materials. One important thing I point out here is The water must be clean, must not contain chlorine. In many cities, we have chlorines in the water in Denmark, and it will definitely kill the plants. I have tried it just to see how does it work, or it will definitely kill the microorganisms it is. I've tried it to see how it is working. The biocomplete extract is used directly on soil and plants. You spray it on. Direct seed treatments. You can put your seeds inside it or spray the seeds with it when you're sowing it to prevent diseases going in. So over here is the same thing, just another picture set up in sort of the farming manufacturing system. Okay, the Biocomplete Compost Tea. So we make the tea, and this is really what this thing here are for, but and it's very much the same as this one here. This is just a small movable system. So an air pump, and then a filter, and so on, made inside. Use the same way as Biocomplete extract. Yeah, you make the tea and look to get the microorganisms to produce, and it is especially fungal and protozoa that multiply. Air is blown into a mixture of 1 liter per minute of water for 24 to 48 hours, depending on the temperature. And it can be used to spray on the plants in the field. So when the plants are already growing, the soil is covered, you can use Biocomplete Compost Tea. And it is to make a biofilm on the leaves. This is what it's all about, that will reject or fight this diseases coming in here. And whether it's bacteria or fungus, this beneficial film this film of beneficial bacteria will reject them. Yeah, okay. There must be the right ratio between fungus and bacteria. There must be the right fungus and bacteria. And this is Dr. Dave Johnson, who's been able to do what we call DNA testing of the bacteria. He has the equipment to see what kind of bacteria is actually growing inside. So in 4 weeks, we have 23 species in the top 80. And this is the best I have done by making permaculture compost. Even though we say we make it in 3 weeks, I have only succeeded to make it in 4 weeks, really. So yeah, the next one is more interesting. This is 60 weeks, as I mentioned before, and we have an awful lot of 99 species, and the very interesting ones. I don't go into all these things because I have to say most of these ones, I don't know anything about them, but they are the beneficial ones in line up. The very interesting ones for me here is the carbon bacteria. It absorbs CO2 from the air and stores carbon in the soil. Very, very important. We have something called the liquid carbon pathway. This is very much these bacteria which are doing this thing. Free living nitrogen bacteria absorbs in from there and put it into the soil. So people think that this, now he's a little bit over the fence, they say. So I put the links in so you can actually go in and see the information of this thing here is coming up and what it can do. Very important thing. Now, next is that the fungus, so we have this bacteria and fungus that are needed in the soil as this basic foundation thing. Plant succession ladder as a function of fungal bacterial ratio, FB. And again, this is Dr. Elaine for Soil Food Web, which have done this thing. But this one very test here is put together by Dr. Dave Johnson again. And here is our crop. Bare soil parent material. We have nothing but our sand, silt, and clay here. And it's very much wheat, because wheat's growing because of the NO3, and it lacks oxygen, anaerobe. So we can see, and it like to grow different kind of microorganisms we are not interested in, as we see in the very beginning. If we can move it from here with the FB to 1 to 1 to, or to 1 to 0.5, move it up here, we get a whole different situation. Here we have a 90 gram production. Here we have a 600 gram production. This is increasing ecosystem productivity. And over here, low carbon, high carbon. So this is what it's all about. We want to get up here. Dr. David Jones have been putting, making this thing as high as over 3,000 grams per square meter, which is quite unbelievable high level, especially when you think that the fastest growing forests in the world are producing 2,200. So he gets higher up by doing these things here right. Very, very productive compared to this. Now we are going to look a little bit at how this thing look in practice. Soil biology succession causes plant succession. So this is our plow field. This is where we want to be, very much with regenerative agriculture. And this is here. This is where we will see permaculture, which are also growing in layers with plants. Layers. They harvest many times a year. It's extremely productive when it's fully grown. So here, this is the one we would like to see 1 to 1. We have bacteria, a few fungi over here, and more fungi, and a lot of fungi to see in the end here. As this is bacteria. This is turned around. This is 1 to 10 in the favor of fungus. But here it will work. Okay. Now, the next one here is really showing the same thing, but what I really want to show with this one here is, again, this parent rock, sand, silt, and clay, we can call it, that we run our farming on, is full of nitrogen. Nitrogen is promoting the growth of our seed weeds, which are coming out every year. This is the first step for nature to recover the land and regenerate the land. This is the seed weeds. So it's interesting that we keep it there, because it's also where there's most work with keeping it. So we move on, and then we get a balance of NO3 and NH4. So nitrogen and ammonia comes up here, and it becomes easier to manage. So this is the ones which are doing no-till and direct sowing, having plants the whole year around inside the fields. Difficult to establish, but if you use the soil food web methods and add in compost, the biocomplete compost and compost extract, etc., then it becomes easier to establish. So here's permaculture and end up over here, and we see even the bacteria, the beneficial microorganisms, are moving all the way over here, and eventually ending up with a lot of micro arthropods inside here. But when we are here somewhere, we have the predatory nematode, the full cycle between protozoa and beneficial, bacteria feeding nematodes and fungal feeding nematodes, the full circle between them. So, yeah, it's working. Let's move on. The biological active soil means carbon in the soil. This is what it's all about. Efficiency of carbon flow into plant biomass. And we are here. This is our conventional agriculture in these two here. So this is the FB ratio again, fungal bacteria. And this is 0.04, which is very, very low and not unusual at all. And this one here, 0.84. If that was a Danish farming soil, it would be very high. I have never seen it that high, honestly. So this is the situation. 3% of the plant photosynthesis is going into actually produce the plant, to produce the crop. This is the situation here. So our conventional farming is here. Between 3% and 17% is going into produce the plants. If we get the FB up to 3.68 or maybe around that, we will move this sort of rate of photosynthesis going into the plant up to 57%. This is also done by this figure here, or this test is also done by Dr. David Jones. And of course, this is a lot more productive, and this is why he can produce more than 3,000 grams per square meter in his field. Next one is maybe something we all know. I can tell that this is how it should be looking almost, and this is what I was told in the agriculture school 40 years ago, that this is what we have in the soil. We probably didn't have it even that time, and we certainly don't have it now. This is way down to 1. This mixture here of fresh residue, decomposing, stabilized matter, humus, and living organisms is likely to be similar. This is how it is indeed. It almost depends what kind of crop you are growing. Organic matter in agriculture soils is often 1%, around 1%, or even below. Before the chemical agriculture was 3.5 to 4.5. There must be over 3% before biology starts work on its own. So what we are adding to the soil with the soil food web method, the whole soil food web, will only be there on its own when we have increased our organic matter, or the plant part, humus, to at least 3% and more. Then it will start to actually appear on its own. The liquid carbon pathway, very, very important thing we go to look at. So we have the atmospheric carbon here and the plant roots here with a lot of mycorrhiza around it. And of course, we know all of this rhizosphere and things like that. The plant roots will grow and develop all of these microorganisms which are dying off, and we have those carbon bacteria, or they store in the soil for hundreds of years, as deep down as the roots and mycorrhiza is growing. Regenerative agriculture and permaculture is not cultivated on a square meter. This is very important. It is cultivated in a cubic meter of soil. So this is what it's all about. Now, all of it, how does it look how does the soil look when we get it to grow? So this is how a soil look when we have no soil food web introduced to it. You can see this is dead. There's nothing whatsoever growing out of this thing. There's a little bit of roots here all the way up in the very surface, and otherwise it's dead. Next one is the penetrometer, which I press into 25 centimeters. I can see this is 30, and it is at 325, 300. This is how far it goes. It's like heating a layer of concrete. No plants are going to grow through there. These people are growing only in the top 25 centimeters of the field. They want to have a soil like that. Now into my own plots. Here you can see it's the same way I dig it up. Just put the fork in and turn it, and then I get the soil up, and you can see plenty of roots all the way into the bottom, and then you can see this loose structure, which is called soil aggregates, which are all over. This is a very poor sandy soil, no doubt about that. And again, the next one is just the same. So this is what we want to do. But the very interesting thing, or at least as interesting as this, is this ones here. I take the penetrometer and I press it right into the ground with just 50 psi, barely register anything. And then it goes all the way down to these handles here, which is about 80 centimeters, and I could even press it further down. I try to dig a little bit to see, and it just moved on into a meter, no problem. And then again, the next one, the grass field, down into a meter. So this is what we are after. We want our root zone, a deep root zone with living soil that is holding a lot of nutrients and holding lot of water and air and things for the soil. Yes. Okay. Thank you very much.